The most common study design for a randomized controlled trial is a two-arm parallel group design. Here, the population of interest, which could be patients with diabetes, are randomized into two groups. The first group gets the new treatment or the new intervention, which could be some new diet with exercise, and the second group gets a control treatment, which could be some form of educational leaflet about diet and exercise. The two groups of patients are followed up to the end of the study, and then an outcome measure is measured on them to test the effect of the intervention. The key element is the randomization process, because by the power of the randomization, all key measurements taken at baseline before any treatment is given on the patients should be equally balanced between the two groups. So their average height, their average weight, their average age, the proportion of men and women between the two groups, the duration of their disease should all be balanced before the start of the study. And this minimises any bias creeping into the study, so that any differences between the two groups at the end of the study can only be due to the treatments given. And that's why the randomised controlled trial is one of the best methods to test new treatments or services. Another type of study design is called a crossover design. Here, all patients receive both treatments and should not know which is the new treatment. They are randomly assigned into two groups. The first group will get drug A first and then drug B, and then the second group will get drug B first and then drug A. So in effect, patients are acting as their own control. And again, measurements are taken at the end of each period to test the effect of the intervention. Usually this type of design is used for patients with chronic long-term conditions, for example with asthma. The drugs should be short-acting drugs because they need to be washed out of a patient's system before the second drug is given. So these study designs can only be used in certain situations. A third study design is the cluster randomised controlled trial. Here, clusters such as GP practices or care homes or hospital wards are randomised to receive the new intervention or the control treatment, which is usually the standard normal practice. Clusters are randomised to avoid contamination of the control group. If everyone in the cluster is receiving the same treatment, then contamination is minimised. The new intervention itself could be aimed at the staff within the cluster with respect to changes in prescribing practices, for example, for infections in care homes, or the individuals themselves through, for example, a health promotion campaign. The issue with cluster randomised controlled trials is that they are costly to run and require a large sample size. So we do need to be sure that this is the best design to answer our research question before we conduct one.